I'll be talking about whether or not number 2 can be number 1, and we'll talk about where that comes from later on. So just a quick outline of what I'm going to talk about, the status of cancer in Canada just briefly. Talk about immunotherapy in the microbiome, the MIMIC trial, which is a trial we're doing for melanoma, and then some of our future pancreatic cancer trials. So just briefly, I'm a medical oncologist here. So basically, I'm a chemo doctor who uses chemotherapy and immunotherapy. I don't really work in a lab. I don't have those skills. And I tend to treat people with GI cancers, including pancreas cancer and melanoma, which is a skin cancer. So generally, cancer in Canada, this is a great figure from the Canadian Cancer Society. It gives a bit of a rough outline of the status right now. This is 2009 data. And the one I'll focus on is the blue circle in the bottom. And that shows the 5-year survival rate for all cancers, whether they're early or late in Canada, roughly in the 2009 era. And 63% of people are alive at 5 years. And that's better than it used to be. But obviously, there's a lot of improvement that's left to go. So another quick snapshot of the cancers that I treat. On the left are the new cases that were estimated for 2019 for men and women. And you can see with my red arrows that melanoma and pancreas cancer are in the top 10 for both men and women. But they're still small numbers compared to larger ones like breast cancer and prostate and colon cancer. And on the right-hand side, it's just showing how many people die or are expected to die in 2019 from the top 10 cancers. And melanoma is no longer in the top 10, which is good, but pancreas certainly is. It's number 4 out of 10, so it doesn't happen very often, but when it does, it's certainly very lethal. And that's something we're trying to work on. So I'll talk briefly about immunotherapy and how it works. So basically the immune system's job, as somebody alluded to earlier, was to kill cancer cells. But a lot of different cancers are pretty good at getting away from the immune system, and melanoma is the best, which is why I've used it as an example. So the big or yellow T cell there, its job is to go and kill the melanoma cell, but each one of them has a receptor on their surface. And when those two come together, it basically puts a pause on the immune cell. So the melanoma cell puts this little receptor out, turns the T cell off, and it can go about doing its business of growing. And we first use immunotherapy in melanoma. And I've drawn immunotherapy as these little letter Ys because they're antibodies, the green ones. And they get stuffed in there, so it stops the two receptors from coming together. And so the T cell doesn't go on pause. It actually can do its thing and go and attack the melanoma cell. And this works fairly well. The survival has done much better over the years, which is why it's no longer in the top 10 of killing people. Unfortunately, it still does far too much, but it's a little bit better than it used to be. And this is what the survival looks like from one of the big clinical trials. And I'll just go through this briefly because there's lots of nice lines. The gray line on the bottom actually shows our old chemotherapy drug over time and how it did. So it's survival. So everybody starts out at the beginning, but the chemo drug did not do very well and people were dying off far too quickly. And then we came up with a new immunotherapy drug about 2011, this green line, and it was better for sure, a lot better, but not quite where we wanted it to be. And then there was an even better immunotherapy drug that came out about 2013-14, the blue line, and people started to do much better again, and it had a lot less side effects. And then when they added the blue and the green together, we got this red line. So that's not so bad anymore. It needs to be up at 100%, but it was getting better over time. And some other features on this that are easier to throw on. This top yellow line is 50%. So that means that 50% of people are still alive out at five years, which is, again, better, not where we want it, but certainly much better. And at the bottom line, the yellow line where chemo used to be, which we don't really use anymore, it was still no better than about 10%. Right? So we went from 10 to 50% survival at 5 years, and we're hoping to get it to 100, but that's a lot better than it used to be for sure. And just to take that figure again on the top, what we notice at the very top here above the red line is that those people don't benefit. They get nothing, right? They take the treatment and unfortunately the cancer still grows and takes their life. The people down below the blue and the red line are this nice light colored blue down here. And those are people who do very well. And they're the ones that survive out to 5 years. And if they do that, the graph starts to flatten out. So they're likely to keep going. And that's great. Up in here where the sort of a triangle is these darker blue people, unfortunately, they don't get very much benefit at all. They get very little. And so we need to do better than that. This figure is also from that same trial, and it's the progression-free survival graph. And it just basically says, how long does the treatment work? So when patients get the first treatment up here, when they're all at 100%, they're all together, By the time they get their first CT scan at 3 months, we have a huge drop off here. So a lot of people didn't get any benefit. The cancer just kept growing and the immune therapy didn't do anything. So that's about 25% have no benefit at all from the combination of immune therapy drugs, which are generally pretty good. And so we call that primary resistance. They get zero benefit, right? And those are the people that we're really trying to help. So now I'll just move on to the microbiome. And as I think most of us know, the human body is colonized by lots and lots of different bugs. We're just coated in them. And there's about 10 to 1 bugs compared to every one human cell. So we're just coated. And those bugs live in certain areas throughout the body. They have like their little villages. And we use the term microbiota, which just means the collection of all those bugs. So you'll hear microbiome, you'll hear microbiota. Here in this talk, they're pretty much the same thing, but they are a little bit different. And so see, these are some of the environments or little villages that the bugs live in. We obviously have lots of bugs in our mouth and in our lungs, on our skin and in our stomach. And we'll focus today on the human gut. So it's got quite a few bugs in it, probably the most of any organ on the body, even more so than the skin, about 100 trillion little bugs. And interestingly, in the bowel, there's almost 1,000 different species of bacteria, which is interesting because I didn't know we had that many, but we do. And you'll hear terms about, a healthy microbiome and a not healthy microbiome. And generally, a healthy one means diversity. So there's a lot of different kinds of bugs. It's not just 1 or 2 types, it's a lot of different types. And we have a bunch of different species, so we tend to call them good and bad, although there's really no bad. But we have a variety of different kind. And then we talk about the abundance, which is how much of the good stuff compared to how much of the bad stuff. And if people are familiar with the C. diff infection, often we hear that as a superbug. That's when there's too much of that bug in the bowel, and that leads to diarrhea, a pretty nasty one. So essentially, the healthy microbiome is like having a balanced diet. You get the good, the bad, and a lot of other things. And the microbiome in the bowel and anywhere else in the body, it's impacted by a lot of different factors. And so there's a few on this picture that I've highlighted. So drugs, antibiotics can kill a lot of bugs in our bowel and change the population. Being ill, there are certain kinds of diseases and illnesses that will affect the microbiome. And then over here on the right, everywhere we live, who we live with, and whether or not we have pets, all those different things in our own little personal environment all have an impact on the microenvironment. So we find that with some people with different illnesses, their microbiome becomes unhealthy. And we see that in cancers, sometimes in bowel cancers, inflammatory bowel disease like Crohn's disease, even in depression and multiple sclerosis. We find that their microbiome becomes less healthy. And what's interesting is that the microbiome actually influences the immune system. It can actually help regulate it. And so generally, if we have a healthier microbiome, our immune system tends to work better. So when we use immunotherapy in cancer, that becomes certainly very interesting to us. And what ended up happening is there was a group that followed along cancer patients who were receiving immunotherapy. And as they were going along their treatment, just before they started, they actually took some of their, a sample of their poop and looked at their microbiome. And they found that people who had a really healthy microbiome or a better one actually did much better on immunotherapy than people who had a bad microbiome. And you can see this over here on the right graph. The blue people are those that had a really healthy one. And so they did much, much better over time. So the question then becomes is, can we turn a not so good microbiome into a good one? In cancer patients, and FMT or fecal microbiota transplantation is the mechanism. And so yes, we can. And what FMT is we take healthy poop from somebody who's healthy, and then we give it to somebody who has an illness or unhealthy poop. And we put it into little capsules. And that's the outer blue one. That is the capsule that people see. It's actually triple coated, so nobody tastes it or smells it. And then we put that into somebody who has an illness or a certain condition. And for us, it's cancer. And so they tried this in mice. And they found that when they gave poop from people who did really well and put it into mice with tumors and then gave them immunotherapy, see how they did. And then same thing with the non-responders or the people with a not so good poop. And what we found was that people with good poop did better, or mice, sorry, that had good poop did better than the mice who had bad poop. So their tumors grew in size much faster. And you can see it out on the bottom as well. They just started to grow a lot. So we found that good poop helps immunotherapy work better in cancer. That's just in mice. And so obviously we need to know if that is going to help for people. So what we try to do is to use the FMT to help people's microbiome get better and hopefully their immune system get better. I just referenced the slide I mentioned before that there's that primary resistance where so many people get no benefit. So the question is can we give them a better microbiome and better immune system, and maybe they will benefit? And that's the hope. And that brings us to our trial that we're running here. The clinical trial is called the MIMIC trial. And basically what we do is we take poop from healthy people and we put that into our melanoma patients, and then we give them immunotherapy to see how they do. And this is the first of its kind in Canada, and we're one of 4 only in the world. Obviously, we started that here with all the great researchers we have here, and we just moved into 2 sites into Montreal so that we can get more patients. And it hit the press last year. It was quite a fun adventure, having all kinds of publicity for something that we're doing here. It went across the world to Forbes in the US, to the Mirror in the UK. There were some African papers that carried it. Very weird experience, I have to say. But the CBC London group had the funniest headline, which was London cancer researchers make number 2 their number 1 priority. And so that's where the title of my talk came from, because we really want number 2 to be number 1. So this is how the trial is set up, and it just goes from left to right. Our patients with melanoma, we do the sampling of their poop and their microbiome right off the bat, and some blood and some urine for some immune cell markers. And we give them their FMT. They just do it 1 time, and it's about 40 pills. And then they get their first treatment with immune therapy. And just before that, we test their stool and look at their blood for their immune markers. And then we keep giving their treatment. And by 3 months, we've taken all the samples that we need to. And then they just keep going with their treatment. And this letter Y up here was like the little green letter Y that I used before. That's what an antibody actually looks like. And we're trying to get 20 patients just to make sure that it's safe. And we've had 6 patients on so far, and they've all been doing fairly well from a treatment point of view. We just added this number 6 for Montreal, so I don't have any numbers, but they just had their poop transplant last week and did fine. We had our number 5 patient just start immunotherapy yesterday and had no trouble with his poop transplant. So all of our people that we've given the poop to have had no troubles. They've been receiving their immunotherapy and they've had no troubles with that. Nothing that we would expect to see or unexpected things where they're more severe. And the first 4 patients have been in for a while. Number 2 has been on for 24 treatments. And they've all had some response. And so partial response just means that the tumor is shrunk, but it's not completely gone away. In patient number 2 and patient number 3, tumors are almost all gone away. So they're doing very well. And so far it appears to be safe. And this is some of the data, which is nice because it's full of bright colors. What we see when we look at poop. So over here on the far left is what the donor's poop looks like. And all these colors represent all the different bugs that are in there. Some good, some bad, some more than others. And the bigger the block is, the more of them there are for each different type of a bug. And so our patient number 2, this is what their poop looked like before we started. And then we gave them the FMT or the poop capsules. And this is what it looked like after that happened. And so it looks a lot different than it did before. And then after we gave them the first treatment, before they got their second, we had another look. And it still looked very much the same or similar to what it had before. So we did make a big change. We don't know if that means anything, but we made a change. And this is just a sample of some of the immune cells that we see changing as well. And these are all the first 4 patients. They're not being compared with each other. They're just being compared with how their immune cells changed after the treatments. So they all started at number 1, which is comparing to themselves. And then after the poop drugs, we see a big change. And then after the immunotherapy, we start seeing more changes. So we know that we're changing their immune cells. And again, we don't know quite what that means, but so far it has been safe, which is the purpose of the trial. So I'll move now to pancreatic cancer. And it's terrible. It's a horrible, horrible cancer. So Dr. Allen had alluded to breast cancer and how it's done over the years. And this is for the 2019 data. And you can see at the 5-year mark, breast cancer survival at 5 years is about 88%. And that's all breast cancers early and late. And for poor little pancreas cancer, it's 8%. It's terrible. And this figure on the right just shows how many people would still be alive out of 100 at 5 years. And this is a very small group. And unfortunately, we all know that Alex Trebek died recently, and he was one of these gray people, unfortunately. So it doesn't matter how much money you have or how much people like you. It's a terrible, terrible cancer. So chemotherapy is not very effective. And that's why there's only those 10 people that are left over after 5 years. And our best chemotherapy for stage 4, people tend to live about 11 months, and that's terrible. And there hasn't been much improvement over the years. And immune cells really can't get into pancreas tumors. It's really tough for them to get in, and that's what these 2 pictures are showing. It's good for our immune cells to be able to kill the cancer cells, but they have to be able to get in. And pancreatic cancer is somewhat more like this, where you've got all these cancer cells behind this really big wall, and these poor little immune cells can't get in. So what we do know from interesting studies, the very small number of people who do really well, they have a lot more immune cells in their tumors. So somehow those cells got in. And you can see here, their survival is more than 10 years for people with really good immune cells inside the tumor. But we tried immunotherapy here in pancreatic cancers, and unfortunately it has done nothing. Because if we can't get the immune cells into the tumor, then they can't go and kill off cancer cells. And so that's the big problem. It's resistant to immune therapy. We do know as well from studies in mice, for example, that the gut bugs, if we give FMT, some of those bugs end up in the pancreas microbiome as well. So we know that we can sprinkle some of them in there. And in some studies, it's about 25% of the bugs we put into the mice gut actually make it to where the tumor is. So the question then is, can we do FMT for pancreatic cancer to make the immunotherapy drugs work even better, like they did with melanoma? So the answer is maybe. And this is where translational research is required here again. So the first part of our plan going forward is an observational study. So we need to look at pancreatic cancer patients and their microbiome or through their poop. And that's our first phase of this study here with some of the researchers in the Cancer Center, one of our immunologists and our microbiome specialists. We're going to be looking at the microbiome of our cancer patients and seeing what it looks like. Because they have this cancer, it's probably not going to be a healthy one, but we don't know. And then we're actually going to recruit somebody from their house, an adult who's considered to be healthy, and see what their microbiome looks like. Because if they live in the same house, they probably eat the same food and have the same pets and do the same things. And so their microbiome should look the same. And there's lots of data to support that. So it'll give us an idea of what pancreatic cancer patients' microbiome looks like, and perhaps what it should like if they were healthy like whoever they live with. And after we give them chemotherapy, we're going to see who does better, whether it's good bugs or bad bugs, if we can even find those. And if the people have a good gut microbiome, maybe they'll do better with chemotherapy. So that's the first start. And then the second stage, basically we're going to be doing it in mice. So we're going to take the poop from people with pancreatic cancer who do well on their chemotherapy, and then those unfortunately who don't do well. And we're going to put those into mice who already have a little pancreatic tumor. And we're going to see what happens. We're going to treat them with chemotherapy and see if good poop mice do better than bad poop mice. And so that's a big unknown. We don't know. That worked in melanoma. We don't know if it'll work with pancreatic cancers. And then the third step is going to actually be doing it into patients, of course. So if we find that it works in mice or there's some suggestion it could, we're going to be doing FMT like we do with melanoma patients, giving them a fecal transplant or these special microbiome-derived products. They're kind of like super probiotics. They're not the regular kind that you can buy. I put those into a patient with pancreatic cancer to try and make their gut microbiome better, to hopefully make the tumor microbiome better. And hopefully giving them chemotherapy or even maybe immunotherapy if we can find some drug company to give us those. We might just see how they do. And so hopefully we can basically take pancreatic cancer patients who don't have a great microbiome, give them a better one, boost their immune system, and then maybe give them immunotherapy and see if they do well. That's obviously the hope, because as I mentioned and showed in that graph, poor pancreatic cancer patients have very little in the way of good options, if at all. And so funding, very important for these kinds of trials. The Hecht Foundation donated some money for us, as well as the Oncology Research Fund. So that was very helpful getting that off the ground. And for our pancreas cancer, we just got some money for the first part from the Oncology Cancer Research Fund. So we're very thankful for that. And I just want to highlight that we have not received any money from pharmaceutical companies. So we're doing this all on our own, which is a testament to the great people that we've got around here to be able to pull off clinical trials with very little in the way of money. So our group here that I want to highlight, Dr. Saman Maleki was a previous RIOT member, and he's the tumor immunologist that's been driving both of these studies for us. Jeremy Burton is a microbiome specialist, and Michael Silverman is the FMT specialist. It takes a whole army of people that are here at the bottom to actually make it work. And we just added 2 hospitals in Montreal, as I mentioned before, and these are the researchers there. And then when we do our pancreatic trial, it's just going to be us here in London, of course, and all the same people are going to help us. So they're a great team. And so, as always, we just want to thank our patients and their families who participated in these trials, because without them, there's no way we could find better treatments for our patients. And before I'm finished, I just want to thank the RIOT team for giving me this opportunity to talk about the things that I like to do. I enjoy talking about it as well and showing pictures of poop.